Yes. So, yesterday in our previous class, we stopped at this stage. We saw how the mitochondrial inner membrane is having membrane proteins, which are called the ETC complexes. And these ETC complexes, the responsibility of ETC complexes is to what? Is to transport electron from NADH to the oxygen. So since the electrons are not directly transferred from NADH to oxygen, they move through a lot of carriers and finally reach oxygen. Fine. So we saw which are the complexes and all the coenzymes involved in complexes. But there was one complex which we did not emphasize, I mean, which we could not start the discussion. That is the ATP synthase complex in this electron transport chain. And in fact, I stopped at this question last class. What is that? While we are discussing the ETC, we are just focusing on the movement of the electron. We are tracking the electron from where it moves to which complex, and then what is the next fate of the electron. We're tracking the electrons. But the oxidative phosphorylation, ETC is a major player in the oxidative phosphorylation process. And oxidative phosphorylation should give us ATP. So how does this happen? How is the flow of electrons in the electron transport chain linked to ATP synthesis? This is the question, right? The link comes now actually. So the link is this ATP synthase complex. And the ATP synthase complex synthesizes ATP by directly combining phosphate, inorganic phosphate group with ADP. It's a direct combination. Please remember, in the substrate level phosphorylation, ATPs were synthesized. But the mechanism was different, where a phosphate group was transferred from much more high energetic molecules to ADP. Is it not? So the kinase enzymes, the phosphoryl transferase enzymes, those things were coming into picture. But here it is a completely different mechanism. The ADP and phosphate group are directly combined. But how much energy is required to make 1 mole of ATPs by this direct combination? It is of course 7.3 kilocalories per mole. But what is the mechanism of this direct combination? How the energy is conserved? In the sense, what do you mean by energy conservation? The energy liberated due to the oxidation of NADH is about 53 kilocalories per mole. How this energy is used to synthesize ATP? That is what we are looking at. And of course, a lot of models were put forth to answer this question. A lot of research went on. And the accepted model today, which is also backed by some evidences, is the chemiosmotic theory of ATP synthesis. And it is not a very old theory, it's a very new one. It was only fully formulated in 1980s, 90s. So, chemiosmotic theory. So, what does chemiosmotic theory tell about ATP synthesis? This chemiosmotic theory basically is basically built on this hypothesis. What is the hypothesis? You know, the potential difference concept, right? The potential difference. What is potential difference? That is also sometimes called EMF. So if you can separate charges, then a potential difference is created. And when the charges are allowed to flow back again, then you have current flowing, is it not? You should somehow create a potential difference. Once the potential difference is created, you can use that potential difference to work, to convert that into work basically. How? You just have to connect the two ends of the, for example, a cell through a wire, and the electrons flow. And you can do whatever work you want. You can use that electricity to make the lights glow, turn a motor, whatever. But how to create this potential difference is the point. And the potential difference Creation requires work, basically. Requires the input of energy. Creation of potential difference requires the input of energy. Once it is created, then fine. You can use that to do work. You can use that to convert that into other forms of energy source. The same thing. And the chemiosmotic theory, what we are discussing is only a very brief synopsis of the extensive theory. Okay, let us at least discuss some fundamentals. Now, chemiosmotic theory assumes like this. So when an electron is flowing from complex 1 to through carriers to 3 and then 4 and finally to oxygen, right? So if this arrow mark is the movement of electrons, every complex, when one electron flows through it, pumps a proton from the mitochondrial matrix, which is in the central region of mitochondria, to the intermembrane space. Okay, so what happens as the electrons flow through 3 complexes? One electron flows through 3 complex. 3 protons are pumped from matrix to intermembrane space. So now if you can look at the charge difference between the matrix and intermembrane space, there is a charge difference, is it not? So the intermembrane space of the mitochondria is rich in 3 protons, whereas the matrix is deficient in 3 protons. So a positive charge potential is created. Where is excess of positive charge? In the intermembrane space. And where is the deficiency? In the matrix. So it's almost like electron potential difference you do in your cells, right? In the cells, batteries you take. One end is negatively charged, the other end is positive. What do you mean by negatively charged? It has excess electrons. So when you connect a wire from negative to positive, the electrons flow, and you can use that flow for that electricity for your works. Same thing happens. Now, once the proton gradient is created, so this is called proton gradient. Once the proton gradient is created, if somehow, if this proton flows back, that flow can be used to do work, is it not? That's what happens in the ATP synthase complex. Okay, so till now, we haven't discussed anything about ATP synthase. So first time we are bringing ATP synthase into the picture. So what is ATP synthase complex? It is basically an ion channel, complex with an ion channel, proton channels. I mean, if you learn higher level biochemistry next, probably if you take up MSc biochemistry and all, you will learn a lot of things about these ion channels in proteins and in membranes. There are something called grated ion channels, non-grated ion channels, all those things. Anyways, don't worry. So protons come from outer membrane, flow from our intermembrane space back into the matrix through this ATP synthase. As the protons flow, the flow of the protons brings about a lot of structural changes and energy. Yes. So you have the proton flow back into the matrix. It creates a lot of structural changes, and as I told you, changes from the energy point of view, and that is used to synthesize ATP by linking phosphate to ATP. So this is only a gist of the chemiosmotic theory. Fine. So just want to repeat it again. As the electrons flow through the complexes, each complex pumps a proton from matrix to outside the to the intermembrane space, and there is a proton gradient created, or simply a potential difference created. ATP synthase is an ion channel protein which allows the protons for the backflow, and as the protons flow backwards from the intermembrane space to matrix, structural and conformational changes which happen in the complex, synthesize its ATP into PI. Simple. But of course, the description of this is very simple. But at the molecular level, a lot of changes have to take place. And in fact, there is one schematic representation of this on Wikipedia, which I just downloaded. In fact, the ATP synthase complex itself is having 3 main parts like this, 1, 2, 3. And there is something called a gamma protein, which rotates basically, in fact. So what is the mechanism of rotation and all is too much. Just understand this rotates. So as and when it rotates, okay, my voice is not clear. Anyway, now my internet will improve. Yeah, if my voice is not clear, please be patient. So as and when it rotates, for the complex, the rotor rotates, the gamma protein. You can see this. See these pink color ones. Pink color ones are ADP plus PI. So ADP and PI comes to the complex, sit on the surface